Hello friends, we have reached to the last week of our lecture series, and today we are going to cover a very important topic in this first lecture of this week, which is metagenomics, which has gained a lot of importance in recent times, and there are so many metagenomic projects going on across the world. So today we will see that how this field evolved, what are the major projects which are going on, what is needed to do metagenomics, how do you collect samples, and we will also discuss some case studies to highlight the significance as well as the applications of metagenomics in the recent times. So in this picture you might have seen many of these things, like traditionally to identify or to study any microbe, okay, we can look it under the microscope, do gram staining and these things, isolate it, culture on the Petri plate, find colonies, and then go for some Sanger identification or some molecular biology or biochemical test, right? But the issue with this is that 98% bacteria cannot be cultured. So if they cannot be cultured, we cannot technically get their genetic information without culturing, right? So we need sufficient amount of DNA for sequencing, and that is why culturing is very important approach. Okay, but since we cannot culture majority of them, there is another way that has emerged, that is metagenomics, which can take care of this. And actually if you see, like in different environments, just to give you an idea, like there could be more than 1 million microbes in just one tablespoon of soil, maybe 1 billion in just 1 liter of water from an ocean. Okay, on your hands you might be carrying microbes which are more than the world's population. In your gut there could be more than 2 kilograms of bacteria, more than 1200 species. So it's a very complex, complicated system, and in fact the number of microbes actually outnumber all the other species that exist on this planet, and there are so many that we could actually name only 0.few% of all of them. Okay, so to address and understand and identify these, we need metagenomic approaches. And not only these environments, there could be some environments such as microbes could be floating in the open ocean, okay, or they may be in the bottom of the sea degrading some organic matter, or in the soil where their composition will change according to the depth of the soil, or a closed system like our gut, where which is like a fermenter which takes some organic matter from our mouth, degrade it, and then you get the excreta. So basically these complex and different environmental systems can only be approached if we have some culture independent method. Okay, and microbes do live in extreme environments also. For example, this is one case where microbes are living in a lake which is actually separated from rest of the earth due to some activity, geological activity, and then when it was found, basically the life was parallelly evolving there for more than 25 million years, and microbes in this environment were actually gaining energy from the radioactive sources, right? In acid mine drainage, which is basically the leftover of once you do the mining and deep mining. So after that, when you leave the mines after the mining is over, water flows through that, rainwater or some stream water, right? And the pH of that water is almost 1. So if you put just an iron ore there, it will melt in some time. So even that environment microbes can exist, right? And in like hot springs or hot volcanoes also they may exist, where the alkaline pH is almost 11 and the temperature could be like very high. So microbes can exist almost everywhere. And just another organismal system, like it's a gutless worm which does not have a mouth, gut, anus or nephridia, even kidney or those kind of organs. And basically it has some of the so basically it has a cavity where microbes are there, which are endosymbionts. Okay, and with some of the starting material when it is getting birth, so those microbes are actually recycling all those material, fixing the environmental nitrogen and actually making it basically making all the amino acid vitamins for this worm that are needed for nutrition, and it survives with kind of a system which is recycling all the waste products and surviving in the minimal amount, right? So microbiome of such a system will be very interesting to know, right? Similarly, our human microbiome, which is now called our second genome, because we have microbes all over human body, and typically in our gut, where they are degrading the organic matter, okay, complex polysaccharides, protecting us against from pathogenic microbes, and basically providing us immunity, and also metabolizing the drugs that we take, the oral cavity or our skin, which is all full of microbes. So basically and actually, like people have now known that they are outnumbered the total number of cells in our own body. So basically they are doing a lot of functions for us. Okay, so basically metagenomics has evolved as a science to explore the unknown microbial world that is cannot be accessed by culturing, right? And the idea of metagenomics is that we have to identify that who all are there, okay, how many are there, their relative counts, and what are they doing in that system, and how they are doing those things in that system. Okay, so and that we can know only when we can identify them, and also get access to their genetic material, identify all the genes in that and their functional contribution to that environment, and also their relative composition. Okay, so what we do in metagenomics is it's a culture independent method, so you don't need to culture the bacteria or clone it or like that. So basically we take an environment that might contain like millions of microbes and like many copies each microbe. Okay, so we extract the total DNA from those microbes, right, and sequence it, fragment it and sequence it, and after that we go for the analysis of those microbes, or the ultimate aim is to reconstruct the their respective prokaryotic genomes. But basically for that you need very deep sequencing and more approaches, but at least the fragments also that we get are informative enough to find out their function. Okay, so metagenomics has two strategies. One, for example, once you have extracted the DNA and doing the sequencing, so one will deal with just with the identification of species, that is using the 16S ribosomal RNA, okay, which is a marker gene present in almost all present in basically all the bacteria, and it is conserved as well as variable. Okay, we will see in the next few slides, and and the last talk of this week also, the third lecture of this week, right. So basically in that we can amplify either the entire 16S or V3 region using appropriate primers, and then either we can now construct their library also, 16S library, or we can directly sequence this and identify the OTUs or amplicon sequence variants and identify the species which are present in that environment. And the second approach, which is basically called the metagenomic approach or shotgun approach, where we fragment the entire genomes without amplifying anything, right. And so basically many bacteria will be in higher number, many in low, so we go for direct sequencing of those, and then do the gene prediction, function annotation, and metabolic pathway reconstruction. Okay, so basically this is the way to access their genetic information and to study them in the way they were in their environments. Okay, the 16S ribosomal RNA gene is a very important marker for the identification of these microbes, because it is present across all the bacteria, and it has both conserved, this green one is conserved, and V9 and V1 to V9 are the variable regions. So it has a combination of both conserved and variable regions, and basically you can design primers from the conserved regions and access these variable regions, and or amplify the entire gene, which is 1.5 kb in length approximately. It varies from species or genus and different species and genus, but this is a very good indicator of which microbe and to identify the microbes. Okay, so basically this is a as we mentioned, we will also cover it later, but 16S ribosomal RNA is a very important gene to identify any microbe in a given environment. Okay, so just to highlight the difference between traditional microbiology and metagenomics. So earlier what we were doing is, from any environment, the bacteria, we try to put them in a Petri plate, and then or basically the bacteria in an environment, you put them in a Petri plate, culture it, okay, like this, identify colonies, and then do gram staining and other biochemical test and metabolic test and all those things. But here in metagenomics, the entire microbial community is sequenced, DNA is extracted. So either they are analyzed using 16S based approach, which is more to identify which microbes are there and their respective composition, or to go for direct shotgun based sequencing, where you will go for the community level genomic analysis, right. So both approaches are used, and we will be dealing with both these approaches in the next two lectures that in this week. Okay. So after the sequencing, it is something like this, that you had a mixture of genomes in hand, which is like a piece of paper. So that is going to a paper shredder, right, and what you got from this is the something like this. So basically you can see these are strips of, right, paper which are like reeds, right. So basically now these reeds are coming from multiple of these kind of pages, right, which actually each page was one genome, and now in these bags the identity of the pages, okay, or those genomes is lost. So you have the challenge is to reconstruct those pages, and also each line basically will have some text written on it, okay, which may be gene, intergenic region, and that will be helpful for your analysis. Okay, so how this field started? Basically it all started with the start of sequencing methodologies, sequencing of the first few genomes, including viruses, important bacteria, right. And then it was mainly done by the Sanger sequencing. Then came the pyro sequencing, right, 454 Roche 454 instrument, that also worked well. Then basically the era of Illumina sequencing came, which is short read sequencing, high identity, basically less error and more reliable, right, high quality sequence, which led to a rapid rise in the metagenomic studies over a period of time, right. And now people are also using long read sequencing technologies to sequence the metagenomes. Okay, and so many studies are going on across the world, including like the first one actually was the Sargasso Sea metagenome, where which is one of the largest one, where Craig Venter explored the metagenome of the Sargasso Sea using the Sanger sequencing technology. Then there is a large scale effort on human microbiome project all over the world in countries, and like joint project by NIH also. So basically that is trying to see that what is the composition of microbiome of humans in various parts of the body, because that has huge implications on the health, and their dysbiosis is associated with several diseases. Then Earth Microbiome Project, where the effort is to identify, understand all the microbial communities that may exist in different environments on this planet, like we have seen some of the environments, right. And similarly, like many populations are also doing their own gut microbiome, like India, and they are also doing, okay, that we will also highlight today. So so basically all these projects are using a combination of 16S based and shotgun metagenomics approach to identify the microbes, and also to understand their functional composition by the metagenomic reads. Okay, so basically for in the case of human, microbes are present in different body sites, skin, and you can see the this pie chart colors are varying in each tissue, like kidney, intestine, stomach, mouth, okay, like that. So they are different in different parts of the body, which tells that their relative compositions may be very different in the different organ sites in the body, okay. And they cover almost all our body, and basically they contribute almost 100 times more genes and functions to humans, which is why they are relevant to us, because we have only 22 to 25,000 genes, but these microbes are contributing 100 times more genes, which perform a lot of important functions which the host or the human cannot do. Okay, particularly in the gut, you have almost 2 kilograms of microbe, more than 1,000 species, which break down your food, protect us against the pathogenic microbes in the gut, protect our epithelial line, okay, by forming a biofilm, stimulate the immune system, which is very important role, and and also metabolize various drugs, and basically their dysbiosis causes a lot of disease such as IBD or Crohn's disease or inflammatory bowel disease or rheumatoid arthritis, and also like cardiovascular diseases, right. So, the factors that shape our human microbiome, or particularly the gut microbiome, could be age, because it changes from when a baby is born. It is said to have no microbes, and it gains microbes over a period of time. Then the microbiome matures and mimics the adult microbiome, and then the old age microbiome. Okay, again, now you are coming from a rural location or urban location, so you may be having a different microbiome. The geographical region from which you are coming, your dietary habits, your lifestyle, your socioeconomic status will all contribute to your microbiome. Then the other factors like stress or lack of physical activity or more physical activity, okay, your diet, okay, diet could be like you are more prone to eat the fat rich diet or a healthy carbohydrate like fiber rich diet, okay. So that will always impact, or long term use of medication, all these things, or some disease or that could have disturbed your microbiome, or some infection. So these things will alter your microbiome. And people have studied the impact of diet on the microbiome also. For example, the high fiber rich diet, which is typically a non-Western diet, whereas high protein and high fat diet, which is typically a Western diet. And so basically people have found that basically the good or healthy microbes are more in the non-Western microbiomes, which is typically consuming the high fiber diet. And the Westernized diet leads to the alteration of the microbiome, and also which is like less healthy and more prone to various diseases. And people have also studied that mouth gut axis, because the microbiome in your oral cavity may impact the microbiome in your gut, and that is a oral hygiene and type of microbes, and the diet that you are taking, okay, will matter when you are talking about the gut microbiome. So this gut microbiome also, when you say gut feeling, basically it is also altering your feeling, mood, behavior, because they are producing a lot of metabolites which can actually reach your brain and can impact your behavior, mood, depression, okay. And they have been associated with autism, anxiety, pain, depression, obesity, definitely cardiovascular disease. So basically microbes are not only degrading your food, but also impacting the host immune system and behavior by their respective composition. So basically, like it could be like this cartoons taken from web. So it typically tells that you might be wanting to have a salad, but your microbiome may be needing more of cheeseburger and fries, your gut feeling. So basically the microbiome really impacts you. So it is, don't take it literally, they will not alter your feelings, but the idea is to avoid the non-healthy food and take more of the healthy food, right. So gut microbiome is also associated with various diseases, because a healthy one will keep you healthy, but if that changes or alters because of any of the things or factor that we discussed, so it can cause diarrhea, allergy, IBD, and cancer, colorectal cancer. It is associated with microbes. So basically they will cause these kind of diseases, and for which like probiotics could be one way to regain, or there are other ways also, fecal microbiome transplant, okay. And therefore we should metagenomics has actually made it possible to explore these things and understand the significance of microbiome, particularly like for the human gut. One experiment, very interesting, they showed that in lean and obese twins, basically one was obese and one was lean, and both were twin. So they took the microbiome and transplanted into a mice. So the lean mice became obese, and from a lean person, when it was given to a obese mice, it became lean. So basically that shows that the significance of microbiome in obesity and in deposition of fat in your body and your metabolism. Okay, and typically in the West, people are suffering with a lot of issues with the gut microbiome, particularly the problem with the Clostridium difficile infection, right? For which what they are doing is they are extracting the microbiome from a healthy donor and converting that into tablets, okay, like these, which can be ingested, or directly doing the fecal microbiome transplant to regain or restore the healthy microbiome, which has also shown very promising results. Okay, but the natural way to improve a microbiome is to have healthy food, more fiber rich diet, and basically a lot of probiotics are available on the shelf, like curd is one example, which when you take in regular intake of that, it will keep your microbiome good. Okay, so basically it is your gut microbiome is what you eat. Basically, if you are eating something like this, it is going to change, become westernized, and basically will be not a healthy microbiome, whereas a regular intake of nuts, grains, fruits and veggies will help you to get a better microbiome, gut microbiome. So we are what we eat. Okay, now when we have looked at the microbiome and some glimpses of the studies that are done environmentally or organismally. So now how to collect the sample? So basically the first step is you should identify that what kind of sample you are collecting. It could be environmental or organismal. Okay, you have to collect all the metadata, like if you are collecting a sample from a healthy individual or a patient. So basically you have to collect metadata including like what the person was eating from the last 3 days at least to maybe 1 month if possible, right, to gain more insights into dietary habits, right, and then record all the other body parameters and whatever is relevant for your objective, right. Similarly, from environment you should note that you are collecting sample from a lake, so what are the coordinates of that lake, geographical location and like that, right. You should fill out those, you should prepare a questionnaire and answer all the questions collecting from either from an individual or from an environmental sample, right. Then you have to collect and that in a sample in a sterile tube or containers or bottles, whichever is relevant, and transport that into the laboratory immediately for further processing or storing, okay, or keeping it under -20. And basically because after collecting from the source, if you take too long to transport and basically take to the lab, it can change the microbiome a lot, right. And at any point of time you should be handled in a very sterile or manner that you are dealing with microbes, right. And you should collect sufficient samples so that you can keep some for future studies, and you might have planned different objectives, and you may need for something for metabolomics, something for metagenomics, something for 16S, something for long read sequencing. So you should have sufficient samples so that you can get sufficient DNA out of it, okay, and find out the appropriate kits and remove the debris and other contaminants from the samples before processing the libraries, okay. So samples could be like any of these environments, open environment, closed environment, right, like this, okay, and water sample, swab sample, soil sample, sediment sample, it could be fecal sample. So a lot of samples could be there for your research. And okay, so basically even air samples and these can also be taken. And when you have identified type of sample, basically you should collect the sample in sterile containers and okay, like these, right, and then use the appropriate metagenomic DNA extraction protocols. Okay, so like some of the ways to collect is like it could be a saliva collector, it could be cotton swabs for collecting the oral swabs. Okay, for fecal sample you can use something like sterile container, okay, with broad mouth so that you can easily collect the feces. Okay, then basically you can see this cotton swab, this part you may touch but still you will use a glove while doing this. and this part is going to collect the swab, okay, and you should not touch this part and directly go inside this, right. Skin sample, it could be the tape strip method or the swab method, okay, or the biopsy. So these sometimes these kind of things may be specialized jobs, and that should be done by authorized persons or competent persons or from vet or doctors, right. So after you have collected the sample, you will transport it, it will go reach your lab, it will stored until you process it for metagenomic DNA extraction. You can do immediately, that is the best. Otherwise you may wait for collecting all the samples and starting together. So it is that decision has to be made, but till then the sample should be stored well, right. Then followed by the library preparation after DNA extraction, okay, and then for going for the appropriate sequencing method, which could be the long read or short read sequencing, and generating the data, and followed by the interpretation of result, okay. So basically again, like it could be 16S based sequencing, where after extraction of DNA you can amplify and amplify the 16S, or going for the shotgun based metagenomics, okay. Regarding sequencing, once you have prepared the library, so basically extracted the DNA, you can go for Sanger sequencing to identify this a particular species of your interest, if you are also doing the culture based approach, okay. But this is not very relevant for metagenomic samples, right. Only when you apply metagenomic culturomics together, then this may be helpful. Then Illumina for short read sequencing, and for basically you can say the deep sequencing approach, and long range sequencing either using Oxford nanopore technology or the PEG biotechnology, okay. Then basically you will be either going for the amplicon based approach, where you will be amplifying some part of 16S ribosomal RNA, which is the hypervaluable region like V3 or V4 or a combination of V3 V4, right. And then after you amplified, you will put the adapters and adapt to this, right. Then barcode, because in one flow cell you can do multiple samples, so to keep some identity for the each individual sample, you will put a barcode to that sample and do the processing, right. On a single flow cell you can do multiple type of sample sequencing. Then the whole genome sequencing, where basically you will be breaking the DNA into pieces and putting the barcodes again according to the sample, and then going for the sequencing, okay. So basically again you are reaching to this picture that you have broken the DNA, and then you have made various generated data from various experiments, sample 1, sample 2, sample 3, sample 4, and then now you have to regain the functional and genomic information for each and every genome, okay. So we might be dealing with the host associated microbiome or the environment associated microbiome, right. So I'll give you some example. For example, the human gut microbiome from India, which was not known, and there are, you can see India is a big country, so many geographical locations are there, and they have different socio-economic groups, urban, rural, diet, gender, okay, all these things vary a lot in India. So you need very comprehensive sampling approach, but still the preliminary studies, or so far the larger studies, have collected samples from all over India, different parts of India, and basically for that the metagenomic approach using the fecal samples, which is non-invasive samples, right, okay. You can apply 16S based or shotgun sequencing approach, and to supplement the results you can also do the metabolomics, including the fecal and serum metabolomics, to further examine the impact of microbial metabolites on the human health. So basically when you are doing this, you might be collecting samples from a population like India, but you also have to do a comparative study with the other population gut microbiomes, because that way you will know that what is different in your population or your set of individuals. It could be healthy or diseased also, okay, and also compare that with some of the diseased gut microbiomes to gain a comprehensive picture of the microbiome that you are dealing with, okay. So basically the outcome of this, because it was done for the first time, increase the information in the international gut microbiome gene catalog, right? And basically lot of unique genes were contributed from India. Our Indian gut microbiome has a lot of Prevotella copri, which could be as much as 90% in some individuals, and which is a very interesting observation, because that means that there is a lack of diversity, or basically there is one predominant bacterial species which dominate our gut, okay, which actually is involved in the metabolism of carbohydrates and so like so this work was published in this paper, and this is one of the very highly cited works from India, right? So once we have known that Prevotella copri is the important microbe from the Indian gut, so basically you can further go for building a database of this Prevotella copri or Prevotella genomes as such, the genus, and then do a to analyze their role in carbohydrate metabolism, right? Similarly, like you are exposed every day to so many microbes or the cosmetics, food, organic matter, right? So basically you apply a lot of cosmetics on skin, hair and other parts, and which might be degraded by the skin microbiome that you have. Similarly, like you are taking a lot of oral drugs, medicines now, nutraceuticals, which are vitamins and those tablets that you take, prebiotic, a lot of fiber rich food, all goes to your gut. So basically, have you ever wondered like what happens to these and how the microbes actually contribute to their degradation? Whether for example, you are using a deodorant that is tested in Europe, on European skin, but when it is applied on human skin of Indian individuals, so it did not show those results, or it was less effective, or also doing some allergic or toxic reactions on the skin. So basically, because of the differences in microbiome, it may happen, and by using artificial intelligence and machine learning approaches, now you can actually predict that how your skin microbiome or gut microbiome will metabolize anything that it is exposed to, because they contain a lot of these microbiomes, a lot of bacteria that have their own metabolic enzymes to metabolize these kind of molecules that they get from either orally or applied on the skin. Like this is one tool, GutBug, which can predict that anything that goes to your gut, you can give the molecule name, and it can predict that what all microbes in your gut can metabolize it, the exact chemical reaction that will happen in the gut, and also what are the resultant metabolites and what species of bacteria will contain that, and whether also that will be toxic to your gut or like that. So basically I am saying I want to highlight that along with metagenomics and basically the big data of biology, the application of artificial intelligence is also gaining a lot of importance these days to use this data to make valuable predictions. Then another like environmental microbiome, if you see, there could be like a very good example is the ocean microbiome, which is huge. 70% of our planet is basically water and covered by oceans and rivers and lakes, right? So you would like to explore, because this will be very surprising and give you new information about the species that exist in those environments. And so many efforts have been done recently to explore the ocean microbiomes, which also vary because the amount of light that penetrate the ocean water to the depth, and basically there could be regions under the sea where there is no oxygen or light, right? So basically it is a very interesting and a diverse environment within an environment, and requires a lot of metagenomic studies. So the first one was the Sargasso Sea study by Craig Venter group, which actually using Sanger sequencing found several new microbial groups, and species also, which are doing a lot of functions in the ocean microbiome. Then happened the Global Ocean Survey, which was done in a 320 kilometer stretch in northwestern Atlantic, and North American coast, and this work also provided very much information about the unknown microbes so far, right? Then recently the Tara ocean expedition, which covered almost all of the earth, including like 68 locations, across the globe, although they left a few locations, okay. And they made a catalog of the ocean microbiome genes and the composition of microbes, right. So now the ocean microbiome can also be like an open ocean. That means you go further down in the ocean, or it could be near the sea or the coastal area where the human activities are more, okay. So these microbiomes will be very different, and the coastal microbiome will tell you like implication of health because it is more exposed to human activities, and also effluents and many other things goes into those kind of microbiomes. Antibiotic resistance could be one of the problems there, right. So these kind of studies in the ocean, and basically this will also tell you about the ocean health, global warming indicators, and impact on the marine species and other life forms, also even eukaryotes, okay. So basically identifying these microbiomes and comparing that with the different regions could be very, very informative. Then like hot springs, which like tag polymerase that you use in the lab, basically was extracted from Yellowstone National Park, right. It can it's a polymerase that can sustain higher temperatures. And similarly hot springs analysis, for example in central India or any other place, can tell you so many other microbes which contain enzyme which can sustain higher temperatures, and which could be important for the survival of that microbe, and also can be used in the lab for various reasons or industrial purposes. So we'll be covering like some of the examples in the later part of lectures also. Okay, similarly like so many rivers are there which are impacted by the human activities, and to measure the extent of those activities, you should measure the amount of antibiotic resistant genes in those microbiomes, or what kind of composition those polluted rivers or water bodies have, and what could be their impact on when we consume those water. So these things become more and more clear when we do such environmental microbiomes, right. And like in a traditional sewage treatment plant, microbiome plays a very important role, because either you are using putting that back into the river, so what microbes are going back to the river after the treatment, or what is happening in the anaerobic sludge digester, okay, or in the treatment using the microbial technologies which can purify the water. So all these things, determination of these microbiomes, right, is very important and critical for the practical and environmental and also applied purposes, okay. Another case could be oil spills, because when you're extracting oil from the oceans, which basically typically host a lot of oil, so there are cases of oil spills. It could be a kind of sometimes accidentally it happens, or sometimes ship spills those oils in the ocean. So that has huge impact on the ocean environment, and in those oil spills there could be microbes which can actually degrade those oil spills. So identification of those microbes it will be very important to tackle such kind of environmental issues in the future. So okay, so one of the study in metagenomic oil spills revealed the presence of so many microbes that can degrade a lot of aromatic hydrocarbons and can be used for the bioremediation purposes. Okay, so basically in this lecture we looked at a very, very important new area of science, which is metagenomics, that covers the environmental as well as the organismal systems, and more importantly human and other eukaryotes also. And we looked at some examples of those studies and typical methods that are used to for these kind of studies. So in the future lectures we will look at the method of analysis that we use for the metagenomic sequences, for the function analysis, and also for the taxonomic identification of microbes in these metagenomes. So okay, this is good for today. Thank you.